Okay, so this mini lecture is about bulk transport, endocytosis, and exocytosis. And bulk transport is the movement of large molecules or large particles either into or out of the cell. So movement of large particles or molecules into or out of the cell. Okay, and this is a type of active transport, which means it requires ATP. Now this is a lot more ATP than, say, a pump protein when we talked about active transport, because now you're actually involving whole membranes forming and reforming. And so if you remember, sorry, my nose itches. Okay, if you remember, membranes, because they're all made of phospholipids, can merge together and form apart, pull apart, and merge together. So this is how this bulk transport, endocytosis and exocytosis, works. And they're basically opposite processes. So endocytosis is going to bring things in, and exocytosis is going to move things out. Oops. All right, next slide. All right, so endo means inside. Okay. And what I want you to see is this involves a vesicle formation. So right here you can see that the membrane is like pulling in and bringing in these particles, and then you form a vesicle. And we've talked about vesicles are these little membrane sacs that can move around in the cell. So that's exactly what's happening in endocytosis. Now, there are three different types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. So let's talk about what the differences are. They're similar if you look at this diagram in that all involve vesicle formation and all are bringing molecules or stuff into the cell. So phagocytosis means cell eating. And phagocytosis is for large particles or large solid molecules. I'm going to put solid particles. And this can be as big as what it's showing you right here, as a bacterial cell being eaten by another cell. Okay. Pinocytosis means cell drinking. So it is bulk transport of lots of fluid into the cell. And yes, there's going to be other dissolved molecules in that fluid, but it's not like it's grabbing one molecule and bringing it in like phagocytosis. It's taking a big gulp kind of the outside environment, again wrapping the membrane around the outside environment and bringing it in. Receptor-mediated endocytosis means that you have a protein called a receptor, and a receptor is a transmembrane protein. So that means it crosses the membrane. And it's very, very specific. So when we talk about proteins, we talk about enzymes, and we'll talk a little bit about receptors. They're very specific, so they can only bind molecules that fit. Okay. And so with receptor-mediated endocytosis, what it's showing you here is that this star-shaped molecule fits. It binds to that receptor, and when it binds to that receptor, it triggers the cell to say, hey, this is exactly what I want, something specific, right? And it brings it in. And you can see that it's only bringing in those star molecules, not the little squares that are floating around. So specificity is the key. Now this is a little bit different because it involves these coat proteins, but it's not something you need to worry about right now. You'll learn more about that in cell biology. The key is you've got large particle eating, phagocytosis, large intake of fluids, penocytosis, and a very specific, I don't know where to put my fingers, a very specific intake of molecules from the outside called receptor-mediated endocytosis. Receptor-mediated endocytosis is one of the ways that viruses can trick a cell into bringing that virus in so that it can infect the cell. Okay. The last one is pretty simple. Exocytosis just means the bulk movement of molecules out of the cell. So exo, exit, out of the cell. And if you look at it, it's just the opposite. You've got a vesicle, right? And it fuses with the cell membrane. And as it fuses, those contents are released to the outside of the cell. So where might this happen? So an example is your B cells, part of your immune system, they produce antibodies. These are really important for an immune response and for protecting you from future infections. And so B cells make these antibodies in bulk. And they're these big proteins that there's no way they could be easily transported across the plasma membrane with just a carrier molecule or a channel. So they make these big molecules and they bring them up in a vesicle and they release the antibodies outside the cell. So then it goes into your blood and lymph system and looks for bacteria, viruses, other foreign substances to tag and destroy. Another example is with neurons. So all those signals and neurotransmitters, and you will learn about those in cell biology, physiology. So neurotransmitters are little molecules that send signals from one cell to another. And so neurons will make a vesicle full of neurotransmitters. That vesicle will bind at the plasma membrane and release all those neurotransmitters to the next neuron so that you can have signals happening. So those are the basics of bulk transport. I've put links to some videos that I think are pretty good. The ones that, oh no, it's not on this one, it's another one. Okay, these are really cool because these are real life. So they are amoebas eating paramecium, so phagocytosis, and white blood cells and macrophages, part of your immune system, also eating bacteria, again, phagocytosis. So take a look at those, they're pretty fun to watch, and the other sites above are good explanations of endo and exocytosis. All right, thanks. Bye.